Bon dia a totes i a tots. Moltes gràcies per vindre. Com sabeu, avui tenim una conferència un poquet especial coorganitzada en l'Institut de Biologia Integrativa i de Sistemes, l'I2SISBio, per celebrant el seu 10è aniversari, almenys del nou edifici, que passa el temps molt ràpid, i tenim l'honor i el plaer de tindre com a conferenciant el professor Szostak. Suppose que so I'm going to change to English. So you know we have Professor Szostak as speaker today, and many of you probably remember last time he was in this room. It was a complete overloaded, overbooking room that up to the point that we had to, well, he offered it in the last moment to give the talk again in the afternoon. So we were talking before because that it was the first time it happened in our faculty and of course in this room, and it never happened again. So we were a little bit afraid about today. But unfortunately, in a way, our students, you all know that they are preparing their tests and their exams and so on, and yeah, they are studying. So we are less people, but I think it's still a great opportunity to hear Professor Szostak, what he has been doing in his, what they have been doing in his lab in the last 10 years, more or less, since his last visit. We can't put exactly the date, but it was around 10 years ago, his last visit. And I hope you enjoy the talk. And I'm going to be very brief in my introduction. Just want to thanks again also our professor Julie Peretó for making the connection and keeping our faculty linked to the big names that show up in town every now and then. So thank you all for coming, and especially to you, thanks for taking your time to stay with us for a while. Thank you, Jack.
Thank you very much. Gracias, Ismael. Gracias, Dega. Thank you very much, Jack Szostak, because of your visit now, taking advantage of the participation in the Jaume I Awards and juries. And of course, just to remind you, this situation 10 years ago, it was a bit funny, but very happy to have so many people interested in origins of life and the lecture by Szostak. Professor Szostak was professor of genetics in the University of Harvard up to 2022, that he moved to University of Chicago. He now belongs to the Department of Chemistry of the University of Chicago. And, well, it is well known because in 2009 he was awarded the Nobel Prize shared with Elizabeth Blackburn and Carol Greiter because of the discovery of telomeres and telomerase, and all the, the science derived of this discovery is amazing, especially all the research related to aging. This was in the first part of his career. I will divide the career in this at least in three sections. This part related to genetics, molecular genetics, and the chromosome structure. In fact, he was the first to design and build an artificial chromosome of yeast that afterwards became also an important tool in genetic engineering. But as many times as he has commented that realizing that the work in that field was a little bit overpopulated, many scientists were moving to this kind of research, he decided to change radically his work. And actually, a few years after the discovery of catalytic RNA, the ribozymes, he decided to start a new adventure related to the origins of life. And particularly, he, we can say, invented a new approach to the origin of genetic information based in vitro evolution of RNA. So he developed, together with other laboratories, all the approaches related to artificial evolution, artificial molecular evolution with RNA. And then, a third section of his career, for me very important and very remarkable, is maybe since 2 decades ago, he started to play with vesicles, lipid vesicles. And he was starting to explore the physics and chemistry related to the behavior of lipid vesicles, and particularly the fatty acid vesicles, and discovering many different extraordinary things that the vesicles were able to do, and also exploring the combination of lipid vesicles with RNA, and to study the RNA replication in the absence of enzymes. So those are very difficult experimental approaches, but illuminating probably essential steps during the early evolution of life and the origins of life. Related to RNA replication, probably he will talk today. The conference or the lecture was announced with a different title, but I like much better this one, the one that is in the slide today, because it's directly related with his latest interest in the origin of information and the manage of mistakes and errors, and that's a fascinating part of the origins of genetic information. There is a recent book co-authored with Mario Livio, an astrophysicist, that is, the title is Earth Exceptional? And this is a book that summarizes very well all the current aspects and views on the origins of life, and also projects to the question of how frequent or not could be life in the universe. So probably at the end, if we have time, we can discuss all these questions. The fact that he has moved to the University of Chicago to lead the so-called Origin of Life initiative, for me is also highly symbolic. When we consider that it was there in the chemistry department to which he now belongs, that 74 years ago, the young Stanley Miller conducted his now legendary experiment under supervision of the Nobel laureate Harold Urey. So Jacques Szostak is a very important descendant of this scientific lineage, and is offering us new views and extraordinary perspectives on the origins of life. So thank you very much, Jacques, again, for accepting our invitation, and the floor is yours.
Okay, thank you. Thank you very much. Okay, so I really want to thank all of you, and especially Julie, for inviting me to come back here after my talk 10 years ago. So it's always a pleasure to come back here and to Valencia. I love visiting the city as well as other parts of the country. As Julie said, my lab has been focused on the origins of life for many years now. And it's a huge set of puzzles, right? To really understand it fully, you have to understand everything from planet formation, early environments, prebiotic chemistry, how this gave rise to the first genetic materials like RNA, as well as cell compartments, and then how early evolution led to the complexity of modern life. So that makes it interesting because we talk to people in so many different fields. But no one person can study all of these aspects. And we have been focusing on a narrow slice of that whole pathway in which we're thinking about what primitive cells, the first cells, protocells, might have looked like. Diagrammed here is a simple cell membrane, probably made from much simpler molecules than our membranes, probably fatty acids or other single-chain amphiphils. We've worked a lot on that, but today I want to focus on the primordial genetic material, which we think is RNA or something very close to modern RNA. And in particular, I want to focus on how that early genetic information, it's a very, very small amount of information, could have been replicated before there were any enzymes or ribozymes to catalyze the reactions. So it has to be, like the origin of life is a problem in chemistry, right? It's the problem of how chemistry gave rise to biology. So in this particular instance, we have to understand how you could do replication just using chemistry and the characteristics of the environment at the time. So it's a very big puzzle. So I'm just going to focus on that. I'll say a little bit about the fact that we also don't know very much about the environments on the early Earth. A large part of the field is now benefiting from the interactions between geologists and geochemists and people who are studying models for prebiotic chemistry in early cells. So the kinds of environments we like to think about are basically what you would find on the modern Earth, in volcanic environments, possibly impact craters. So these images are taken from Yellowstone National Park, which is a large volcanic area with many things that lead to fluctuations in temperature. You have these outflow channels that give you exposure to ultraviolet radiation on the early Earth, but also environments like this alkaline carbonate lake in British Columbia, which is a geochemical setting that can lead to the concentration of phosphate, which we know had to be present and available in order to make nucleotides. So in some way that is still rather mysterious, these different environments probably led to different parts of the prebiotic chemistry that gave rise to the building blocks of biology, and eventually they had to come together so that these molecules could self-assemble into protocells, and also so that the chemistry and the environment could lead to the replication of those primitive RNA genomes. So that, again, we can break down into a series of steps, right? So there's a whole field of how to make the building blocks of biology, and especially nucleotides. A lot of this work has been done by my colleague John Sutherland in the UK. How they polymerize into short oligonucleotides. Several people have worked on this. There are many theories being explored. The one I like the best is once you have activated nucleotides, if they just go to a low temperature so the solution freezes, These molecules get concentrated in between ice crystals and they will polymerize spontaneously. So this was discovered by accident by putting a solution in the freezer, which we usually do to prevent anything from happening. And then you come back a few weeks later and you see long RNA chains. So it can be a very simple process, but this is the kind of thing that tells us what environments might have been necessary for some of these steps. Okay, so once you have these short oligonucleotides, then we can start to think about how those sequences could be copied and then ultimately replicated, and how the heterogeneity of the chemistry could have been overcome, giving rise to more homogeneous RNA molecules that would be a better starting point for Darwinian evolution. Okay, and then we have to go actually beyond just copying a template to actually replication cycles, which also brings in a whole new set of challenges. So today I'm going to talk about template copying chemistry and then how you would go to replication. And a large part of that has fascinated us recently is the accuracy of the copying. It's not enough just to copy a template if there are so many errors that you can't transmit any useful amount of genetic information. So the fidelity of the copying chemistry is a really critical aspect that's turned out to be more complicated and more interesting than I ever expected. Okay, so I'll start by talking a little bit about the basic chemistry of copying an RNA template. And that also turned out to be full of surprises. In fact, everything in this whole field of origin of life has turned out to be full of surprises, which is one thing that makes it so interesting and fun. But let me begin by saying how we thought it should work, which was based on our preconceptions from how things work in biology. So we knew from the work of the late Leslie Orgel that the substrates had to be much more reactive versions of nucleotides. So typically phosphorimidazoids like this, much more reactive than the NTPs used in biology, okay, because there are no enzymes. So these are reactive enough to polymerize spontaneously. And the basic system is like this. We have an RNA primer, base paired to a longer RNA strand. This is the template region we want to copy. And the idea was these activated monomers would come in one at a time, bind next to the end of the primer, react with the 3' hydroxyl, and extend by one nucleotide. Right? Exactly the way things basically work in biology, except with NTPs and catalyzed by enzymes. So it turns out, though, that this kind of reaction is, if you only let one monomer come in, it almost doesn't work. It's incredibly slow, and the fidelity is really terrible. And it turned out that actually Leslie Orgel's group had found now, I think almost 30 years ago, that if a second monomer can bind downstream of the first, all of a sudden the reaction is much more effective, much faster. And the mechanism, though, remained unknown. Orgel's paper was in the literature. I think almost nobody read it. Certainly nobody followed up on it. A student in my lab, Noam Privis, rediscovered this effect independently. And after working in the lab for many months, characterizing this effect, he realized, oh, it's all been done already 30 years ago by Leslie Orgel. But then we decided, okay, this catalytic effect, right, this effect of this downstream nucleotide seems to be critical for getting the chemistry, the copying chemistry, to work. So we decided we really have to figure this out. And it took us several years to get to the bottom of it. We tried all kinds of different approaches. We assumed at the beginning that maybe this catalytic effect was sort of by analogy to how enzymes work, some conformational influence that would restrain the upstream nucleotide to be in the right conformation to react. But we could never find any evidence for that. We tried all kinds of different approaches, structural approaches. Nothing worked until eventually a new student joined the lab, Travis Walton, and he actually figured out what was going on, and it was extremely surprising. What Travis figured out is that two of these activated nucleotides, and now here we've switched to a slightly different activating group, these react with each other to make dinucleotides that look like this. Okay, so they're bridged by this imidazolium group between the two 5' phosphates. And this makes these bridged dinucleotides are what are the actual substrates or intermediates in the reaction. And they're better for two reasons. First of all, they can bind to the template by making two Watson-Crick-Franklin base pairs. And also, when they bind to the template, this bridge structure is organized in 3 dimensions to favor the inline attack of the 3' hydroxyl at the end of the frame. So the reaction goes much faster. It also goes more accurately, and you need much lower concentrations of these substrates because the binding is much tighter because there are 2 base pairs. So this reaction mechanism was controversial for a while. Even people in my own lab didn't all believe it, because how could something fold and bind by 2 base pairs like this? And but then the next step in figuring this out came from structural studies that were done by Wen Zhang. Wen was a postdoc in my lab. What Wen was able to do is grow crystals of short RNA duplexes with a little overhang that would be a template region. He would grow the crystals with an unreactive nucleotide bound to the template, harvest lots of crystals, and then soak the crystals with our activated substrates. And after soaking for 15 minutes, he could see that these activated monomers had gone into the crystal. They're base paired to the template, which is up here. They're stacked on the end of the primer. This is the last base of the primer. But the leaving groups, the imidazole leaving groups, are completely disordered, so we don't see them. They're just unconstrained in the solution. But amazingly, when he allowed the crystals to soak for an hour and then harvested them and solved the structure, you could see that there's new electron density between the 2 phosphates. So this is the formation of this imidazolium bridged intermediate in the crystal. And again, 2 perfectly good Watson-Crick-Franklin base pairs to the template. And then, remarkably, if you wait even longer and add the catalytic metal ion, you can see that this density has gone away, and now there's new density between the end of the primer and the incoming monomer. So he's seeing all steps of the reaction happen in series in the crystal up to the formation of a new phosphodiester bond. And so that really convinced us that this is the relevant kind of chemistry. There are some details of the prebiotic chemistry that make, I think, a very strong argument that this could actually be the real chemistry that was going on at the time of the origin of life. And I can talk about that later if there are questions. But for now, I want to move on and talk about how we've studied this reaction in terms of actually copying templates and looking at the whole process. Okay, so for decades we've studied these reactions by doing reaction kinetics on a single template, taking time points, running the products on a gel, seeing what gets made. It's very slow, very tedious, and you can only look at one thing at a time. And then Daniel Dosdovich joined my lab 7 or 8 years ago now, and he sort of dragged us into the modern era and showed us how to use next-generation sequencing to follow these reactions in a massively parallel manner. And the basic idea is it's very simple. We make a hairpin RNA, and this part of the RNA is the template that will be copied. When we add our activated monomers or our bridged dimer substrates, the 3' end here gets elongated by copying the template. We let the reaction go for a while. Then we ligate on the tags, the oligos that you need for Illumina sequencing, amplify everything by RT-PCR, put it into the Illumina sequencing pipeline. We get millions of sequence reads. And so from those sequence reads, we can see that we have molecules with every possible template sequence, and every template sequence is linked uniquely to the nucleotides that got added at the 3' end. So we see the products for copying every template. We see how far it went. We see the mutations that were made. We see the context in which errors were made. And this has been a remarkably fruitful approach, which has been the basis of most of the work that I'm going to talk to you about for the rest of this talk. Okay. So the first time Daniel did that experiment, the results were kind of shocking and disappointing to us. So what you see here is the nucleotide that was added, A, C, G, or U, at the first position of copying the first template base, the second, the third, and the fourth. And what you can see is that the distribution of added nucleotides is extremely biased. C and G are copied well, C too well. The A's are difficult to copy, so after copying for even 2 or 3 nucleotides, there are very few A's being incorporated. And for U, it's the worst. The addition of U's is almost completely gone, even by the second nucleotide extension. So this showed us immediately that there's something wrong with this system, right? This could never be the beginning of the origin of life because you couldn't transmit any useful information with such a biased incorporation of nucleotides. So we knew that there had to be some kind of solution to the problem. And so that's, let's see, what we've been working on quite a bit. It turns out, I think satisfyingly, there are actually several possible solutions. Okay, so the first thing that Daniel tried, actually, was to make the system more complex in a way that could mimic the complexity of the environment in early life. So instead of having just mononucleotides and the bridged dimers that they form spontaneously, he also put in random sequence oligos along with the activation chemistry so that those oligos could also be activated, react with monomers, make monomers bridge to oligos. Then these longer structures could bind to the template. The idea was if you have a short oligo, it could hold down something like U long enough to actually react. Okay? And it worked spectacularly well. So here again, we're seeing the product that's incorporated at the first, second, and so on, nucleotide of the template. And you can see it's much, much more even. Even the worst nucleotide U it's less efficiently incorporated, but it's still present at a high level throughout the copying of the whole template. So that was very satisfying. That was one solution. But we've been exploring other possibilities. And one of the other attractive potential solutions comes from thinking about the primordial genetic alphabet. So was RNA always A, U, C, and G? Or could there have been earlier versions of that alphabet? And one of the reasons for thinking about that comes from the prebiotic chemistry leading to the synthesis of nucleotides. And in the pathway that's been explored by the Sutherland group, the immediate precursor of U is 2-thio-U, shown here, and the immediate precursor of C is 2-thio-C. And this is very interesting because we had been studying 2-thio-U already for many years because it makes a more stable, a much stronger base pair with A. 2-thio-C makes things a little more complicated because it actually makes a distorted and weak base pair with G. However, if you make it base pair with I, with inosine, this base pair is almost exactly the same shape and the same stability as the 2-thio-U-A base pair. And furthermore, I, you can make simply by deamination of A. And so we've been exploring this potentially primordial genetic alphabet. Our thermodynamic studies show that these base pairs are completely equivalent. And so when we tried to do the same kind of sequencing experiment, again, the results, it was very satisfying. So you can see, and now again, all 4 nucleotides continue to be incorporated as the copying chemistry goes on. Now, I have to say, we haven't published this yet because we're not quite satisfied with the technical details of copying these modified nucleotides. So we're still working on it, but I suspect that this is more or less correct, but we're still working on it. Okay, it turns out there are also other possibilities. So a simple thing to look at is just environmental conditions, like the temperature. And it turns out that if you go to much colder temperatures, that helps a lot. Also, A is now copied reasonably well. Even U is not eliminated. It's still biased against, but not so bad. On the other hand, if you go to higher temperatures, that makes things much worse. So maybe this is telling us something. Maybe this is actually saying that the environment where this kind of copying was going on early on had to be cold at least some of the time, because that would make things work much better. We've tried other simple solutions. For example, all these experiments, we put in the same amount, the same concentration of all 4 nucleotides. And so we thought, we can fix the problem. We'll put in more U and maybe less G. That made everything worse. Everything we tried to do by changing the ratios to try to fix one problem causes a new problem. So I think we're kind of focusing in on conditions and chemistry that would allow more or less unbiased nucleotide incorporation, which is important because it allows the exploration of more of sequence space and therefore the evolution of more interesting ribozymes that could do useful things for the cell that it's in. Okay. Let's see. Where are we? Okay. So far I've only talked about copying a template and making sure that all 4 nucleotides can be incorporated. But as I said at the beginning, just copying a template is not good enough. The accuracy is super important. Okay? And so one of the first things that we learned from the deep sequencing approach is that the accuracy of our copying is just terrible. Right? It's so bad that you actually couldn't possibly transmit any useful amount of genetic information. The low fidelity however, has turned out to be also very surprising. We expected, again, based on what we see in biology, that the wobble base pairs, the GU types of errors, would be the dominant error. It's totally not. We were very surprised to see that the incorporation of G opposite A in the template is by far the dominant form of mismatch. And we still don't fully understand why that is. And you can make up explanations because G is stronger stacking interactions, but we don't really know. Okay. So let's see. The basic problem is that the error frequency in copying one of these random templates is about, in this particular experiment, is about 11%. It varies from experiment to experiment depending on the conditions, but it's usually in the range of 10%, can be worse, can be a little bit. That means that you could only transmit a genome with about 10 important nucleotides, right? So this is worked out mathematically by Manfred Eigen decades ago. The idea is that the so-called Eigen-error catastrophe. This means that no matter how strong the selective pressure, if the error frequency is at a certain level, the maximum genome size is roughly the reciprocal of the error frequency. So that means if the error frequency is 10%, you can transmit 10 nucleotides worth of information, which is not enough to define any kind of useful RNA structure. Okay. We would like the error frequency to be more in the range of 1 or 2%, because then we could easily encode and transmit the information required to define at least a simple ribozyme that could do some useful function. Maybe be a ligase, a primitive polymerase, maybe an exonuclease that could chop out errors, for example. Something like that. So the question is, if the chemistry is giving us error frequencies in the range of 10%, how can we possibly find conditions that make it work 10 times better? So that's what we've been working on a lot in the last couple of years. And I'll tell you again, we've found some very surprising things that make this whole thing much more interesting than I ever imagined. Okay. So the first thing, again, is temperature. So again, just as going to low temperature made the copying less biased, it also improves the fidelity and brought the overall error rate down to about 6%. It's still pretty terrible, right? You still can't transmit more than about 15 or 16 nucleotides worth of information. So we need to do better than that. Interestingly, though, going to higher temperatures is terrible, right? So again, I think it's nature is telling us something that the copying had to be probably had to be at low temperature early on. Okay. So let's look into that in a little bit more detail and compare the low and the high temperature. So this is a little too much data to absorb, so let's break it down and look at what happens when we're copying a C or G. So in the template strand, we want to copy C or G. We ask what gets incorporated in the growing primer. And here, the fidelity is pretty good, right? In both cases, the errors are only a few percent at low temperature. Clearly much worse at high temperature. Okay. So it turns out the problem is mostly when there's A in the template. When A is in the template, it's very hard to copy it accurately, and you can see that here. So again, if we focus on A in the template, at high temperature, you can see it's almost random. There's almost no fidelity. At low temperature, the error frequency here is still about 20%, 20 to 30%, depending on the experiment. So why is that? It turns out the context also plays a big role, and we can see that by analyzing the sequences in a slightly different way. And this is, again, to my surprise, I thought we were studying simple chemistry, but it turns out that bioinformatics has been central to understanding what's going on here as we analyze these millions of sequences. Okay. So in these experiments, we consider the particular template base being copied, and then we ask, what happens, how does it depend on the next template base? Okay. And this matters because, remember, the substrates are actually dinucleotides. They're these imidazolium-bridged dinucleotides that bind to the template by 2 base pairs. So again, let's break this down and look at this when the next base is a C. Then the copying of C and G is really quite accurate. U is not too bad. A is again the worst, but now the error frequency is less. It's about 10% here. Okay, so it's an influence of the next base. If that's binding more strongly, the substrate is probably more well organized and the fidelity is higher. If, on the other hand, the next base is A, so you're trying to copy an A, and the next base is an A, then you can see that the error frequency is super high. The correct incorporation of U is less than half. Right? So there's a very specific problem in copying sequences that are AA. Okay, so what to do about this? So we thought maybe if this was really what was going on at the beginning, any genome that had sequences like AA, and therefore also UU, right, from the other strand, but maybe AU and U, maybe they just don't work. So they get filtered out. So we can computationally remove all templates that have strings of A's and U's. Right? And only isolated A's and U's. And ask, what is the fidelity then? And not surprisingly, that does help. Okay. So we just, as I was saying, if these don't work, they just die out. So we can delete them in the computer and ask what happens. And again, the fidelity when we filter out these problematic sequences gets somewhat better. And so now, with a combination of low temperature and filtering out these difficult sequences, we have an error rate a little bit under 5%. Okay, so it's still, it's better, but it's still bad. Right? So what else could contribute to higher accuracy of copying? Okay, and so here we come to 2 surprising things that actually make a huge difference. Okay, so the first effect is post-mismatch stalling. This is a very simple thing. It's actually universal in biology. When you're copying a template and you make a mistake, it's very hard to keep going. Right? The geometry of the mismatch is such that catalysis of the next step is extremely slow, generally, depending, again, on the sequence. Okay, this was initially looked at in a chemical system by Irene Chen, and she found that stalling can be a quite large effect. So Orhan Asikaz, a new student in my lab, has looked at this by the sort of our old-fashioned classical methods of doing reaction kinetics. So he has a system where he can vary the nucleotide here, N1 and N2, the end of the primer and the corresponding base in the template. So he has all 16 combinations, and he says, what is the reaction rate when the substrate binds to the template here? Same substrate, same template, just different correct or incorrect base pairs. And what you can see when we compare the reaction rate to the correct, so if the correct base pair here is U paired to A, call that a rate of 1. If it's a G, it's 5-fold worse. If it's a C, it's more than 100-fold worse, and so on. So you can see copying after every mismatch is much worse, except for the wobble base pair, GU. And surprisingly, it's very asymmetric. So UG is a strongly stalled, GU is not stalled at all. Okay, so that's what we see from reaction kinetics. And so then the question is, how does that influence the fidelity of products that have continued to get copied? Okay, so the basic idea going into this was if the copying has continued, it's the molecules that get the furthest are the ones that by chance didn't make a mistake. Okay? If there is a mistake inside, the probability of continuation would be much lower. So we can look at that by sequencing. And just show you one example. So this is from an experiment very similar to the first one I showed. So here the overall error rate is 12%. And the mismatches, the errors, are shown here. Again, the dominant one is G on A, also U on G, and others contribute. But now, if we look at sequences where the second base is correct and asked what happens in the first base, right? These have been filtered out because if you have these mismatches, they don't keep going. So if you look at the subset where +2 is correct and ask what happens to +1, there are fewer errors. The wobble is enriched, and the overall error rate is now less than 1%. So because of this stalling effect, the sequences that do manage to get extended are extended much more accurately. And now we have an error rate below 1% that would allow for the inheritance of enough information to code for useful structures. So this was very exciting. We're still trying to understand why this happens. So we're doing both crystallographic experiments to get crystal structures of mismatches and see why the reactions are slower, molecular dynamics simulations, and so on. Okay. Let's see. So, as I was saying before, it's not enough to just copy a template once. You have to copy the copies, right? We need to have an ongoing cycle of replication. So how do you do that? And it turns out that is actually a problem that people have been frustrated with for at least 60 or 70 years. And it was so difficult that actually nobody wanted to think about it. We didn't even want to think about it because we couldn't imagine any solution. And there are many problems, but the big one is that if you copy some long RNA strand, it's very hard to get the products apart. Right? The RNA duplex is so stable, it's hard to get strands apart. Even if you can, if you return condition to conditions that allow base pairing so that you can do copying, and then the strands come back together much faster than the copying chemistry. So the strand separation problem is hard to imagine a solution to for a long time. And there are other problems, such as how do you copy the beginnings of both strands? Because you would need defined sequence primers, which would not be available early on. Okay. So we didn't even want to think about this problem until during the pandemic. Like I guess all of us, there was a time we couldn't go into the lab. We had to stop doing experiments. We were just sitting at home, nothing to do but think. And so during that period, we came up with the model shown here, which is called the virtual circular genome model. And the idea is the genome of a primitive cell, of a protocell, would be a circular sequence, but no actual circular molecules, right? It's not like you would do rolling circle replication. The idea was that instead we would have a large collection of fragments, short oligos that map to both strands of a circular genomic sequence. Okay? And it's the collective interaction of all of these short oligonucleotides that allows replication to happen. So every oligo can act as a template or a primer or a helper and so on, and that's shown here. Right? So if conditions favor base pairing, these oligos will base pair with each other. Some configurations don't allow for any copying chemistry to happen. Some configurations allow a little bit of primer extension. And then you do that, then you raise the temperature or lower the salt or something or change the pH. Everything dissociates. And if you have a cycle like this, every time you do base pairing, you allow base pairing, you can do a little copying at random positions all the way around the genome. Right? So it's completely different from copying in biology, where you start at the beginning, you copy around until you get to the end. Very processive process. Here, the copying is completely distributive. So it's like a massively parallel process for copying chemistry. So we're studying this by a combination of experiments and theory in collaboration with the Murugan lab in physics. But let me show you one of the most interesting implications of this model that was actually pointed out by my colleague Arvind Murugan, who's in the physics department at UChicago. And Arvind pointed out, if you consider a random genomic position, right, the nucleotide at this position is represented by a family of oligos that overlap this position from both strands. And so this position can be copied into the opposite strand by many primers that can bind downstream of that position. Okay? On the other hand, if you consider a new mutation, as a mistake has been made here, right? This position, this mutation cannot be copied into the complementary strand at the beginning because there are no primers that can bind to anything downstream of this mutation. So it can't be copied into the genomic cycle until it gets extended on a template. And because of the stalling effect, that's a very slow process. Right? So there'll be a delay before any of these new mutations get incorporated into the replication cycle. And through some model systems and some computational studies, we think this could give potentially another tenfold increase in the fidelity. And that would make the overall replication frequency somewhere in the range between 0.1 and 1%, which would allow, in a purely chemical system, the transmission of enough information to encode multiple ribozymes. Okay. Let's see. I think I will... Oh, yes. If I have a couple minutes, I'll tell one final little interesting thing. This is really more of an idea at this point. But if you think about what would be going on in a protocell with this chemistry which is giving rise to oligos now that terminate in mismatches, they are accumulating because they have difficulty being further extended. That's a load on the system, right? What is that going to do to the whole replication system if you have all of these oligos accumulating with terminal errors? And so this is just an idea for how those oligos that we assumed would be a bad thing, right, that might even lead to the death of a protocell if there are too many of them. Maybe it's not so bad, or maybe it could even be a good thing, a useful thing. And the idea comes from a reaction that we've studied for other reasons entirely, called loop closing ligation. The idea is if you have two oligos that are bound to each other by base pairing, but one has an overhang, then if the 5' phosphate here is activated, the 3' hydroxyl here can attack, and you can make, in one step, a stem loop RNA structure. So this is forming a stem loop, and it's not directed by a template. It's just an intrinsic property of having oligos interact with an overhang. Okay, now imagine if, well, the ones that accumulate here and that can do this will be preferentially the oligos that end in a mismatch. And those, we've shown, can still do loop closing ligation. Okay, so the first implication of loop closing ligation is that it can sequester oligos that end in errors. So instead of sticking around until they eventually get extended and could cause problems, they get filtered out of the system by being turned into these hairpin loop structures, which basically could not be replicated. Then we thought, well, actually, maybe this could even be a good thing. So the mutations, a lot of, in biology, the stem loops that are the most common and most stable are the GNRA tetraloops. The closing base pair is a GA mismatch, which is the dominant mismatch that we see in our chemistry. So maybe those GA mismatches that for years we've been trying to figure out how to get rid of them, maybe they're actually a useful thing because they help to build up structured RNAs by taking part in loop closing ligation. So this is a hypothesis that we're studying now, but we've seen in many cases that things that we thought were really problematic have turned out to be useful in the end. So we're still exploring this, and we'll see what happens. Okay. So I think at this point we're okay. So that's what I just said about making these stem loops. I think we're really approaching a situation where we're ready to try to actually make this cycle work. We already know how to do vesicle growth and division spontaneously. We've worked on that for many years. We're now at a point where I think we know how to do the replication chemistry and to do it with sufficient accuracy. So the next step is to try to put everything together and do many, many generations of this kind of growth and division and genome replication using prebiotically reasonable chemistry. And the hope is to actually watch the beginnings of evolution happening in real time and see what actually might have been the first things to evolve in early life. So we're very excited. This is what we're going to be doing for the next few years. And I just want to end by thanking at least some of the people who've been involved. I've tried to mention some of the key names as I went along. These are on the top row are all people who've gone on to positions either in academia or in companies, and these are some of the current people in the lab working on these problems. A lot of the sequencing work has been done by Yan Feng and Ben. And you can see, and I should also mention my collaborators, are Urban Murugan and people in his lab who have helped us with the modeling of this kind of replication scenario. And I just want to end by saying you can tell just by looking at the pictures of all of these people that the lab comes from people who have come from around the world, right? We really try to attract the best people from everywhere, Europe, India, China. And I think that's one of the things that really drives science forward, is just getting the best people from everywhere. And, fortunately in Europe, I think that is still the way things work by and large. It's very unfortunate in the United States that we're now trying to push these people away. But I'm hoping that phenomenon won't last too long, and we can get back to a more sane situation and celebrate science the way it's celebrated here. So thank you. Happy to answer questions.
Yes, thank you for the talk. So we have time for questions.
Thank you for your presentation, Professor. I guess all you have been explaining happened around 3.8 billion years, more or less. But my question is, why did it happen only once in 4 billion years?
So, of course, we don't actually know that it happened only once. What we know is that all life now traces back to a common ancestor, but there was possibly up to 1 billion years of evolution before that, right? So there may have been many lineages, many branches of the tree that all went extinct after the actual origin of life. But at the beginning, we don't know if it was very easy for life to start once conditions were good. Maybe life was popping up all over the planet and even exchanging information as part of early evolution. Or maybe life is very, very hard to get started, very extremely rare process, right? In which case, maybe it did only happen once. But we don't actually know. I mean, one of the things we would like to get to in understanding is it potentially very easy for life to get started, in which case the universe may be populated with life everywhere? Or maybe the origin of life is so difficult that we're the only place in the universe that has life. We don't know, right? That's one thing that's one of the big questions.
Hi. It almost seems that the original genetic code had only 2 letters, C and G, because are the more stable, the ones that get copied with more accuracy. It is curious that with only 2 letters, in principle you could do 8 amino acids, but you only do 4 because the third position is irrelevant. Could this be related somehow?
Yeah, I don't actually think that you could get started with only 2 nucleotides. There have been many experiments that have been done by directed evolution to try to evolve aptamers or ribozymes made from only 3 or even only 2 nucleotides. The problem when you have only 2 nucleotides is you form many possible secondary structures that have almost the same energy. It's very hard to find sequences that are focused on forming one structure. I think starting with only 2 is almost impossible.
In the meantime, I'm going to ask. So you mentioned when you are copying a template that the next base was important, but I guess that what if you continue with that? So what is the long correlation? So what about the next one also?
Right. So we haven't done that yet. Obviously, there could be influences that extend further, and so eventually, especially if we're using monomers bridged to oligos, I think that becomes really important. So I think with enough sequencing experiments, we will eventually get to that. We can also look actually going backwards. So as I said, copying after a mismatch is very slow for most mismatches. A mismatch 2 nucleotides back also has an effect, a smaller effect. And even 3 nucleotides back has a measurable influence on fidelity. So we interpret that as being the geometry of the duplex is perturbed by mismatches over at least 3 nucleotides, 3 base pairs.
Hello, thank you for your great work. I have a question. When you are testing the errors, I guess that you start with the same amount of each of the blocks.
Yes. Yeah.
Probably is the best approach, but maybe it's not
Right.
near to the reality.
Right.
Do you think that if you use more amount of the less probable letters, can you rescue the problem?
Yeah, so we tried that. That was one of the first things we thought. There's no reason why all 4 nucleotides had to be present at the same concentration, right? It seems completely arbitrary. So we have tried playing around with lots of different relative concentrations. The first thing we tried is, okay, U is difficult to incorporate. We would just put in more U. So it partly solved that problem. But then we got a huge increase in the UG mismatches. So we solved one problem, we created another problem. And everything we've tried has been like that. You solve one problem because of something else that can be worse. So I'm actually more optimistic that using 2-thio-U might solve all of these problems, but we're still in the middle of those experiments.
And I have another question. I want to know if you try to replicate some extreme environments that can be more related of the origin of life. I mean, high pressure or high temperatures.
Yeah. Yeah. So I showed some of the experiments where we looked at moderately higher temperatures, and everything is worse. The bias is worse. The mutation frequency is much worse. So I think the simplest interpretation is that cold environments are better. I think there's a common view that the origin of life might have taken place in extreme environments, because there's this myth or misunderstanding that the early Earth was a horrible, violent, hellish environment, but it's actually not true. There were impacts, of course, but most of the time it was, I think, quite normal environment. Only periodically you would have large impacts that were very destructive. But there are millions of years between those events. So I think that the origin of life is something that actually happened when the environments were very moderate, very temperate. Everything had to be just right to let life get started. And then as it evolved, it learned to adapt to more difficult conditions and ultimately to the extreme environments that life has adapted to today, and as you see from the various extremophiles. But I think that it's just my opinion, but I think the origin of life was restricted to very moderate temperate environments.
Taking into account the environmental factors that increase the fidelity of the copying and also the necessity for phosphate groups and activated nucleotides for all of this process, do you have any idea of a point in time or a place where all of these conditions could meet at the same time? At the same time?
Yeah. Well, I think there are very few constraints, right? I mean, we know after the moon forming impact, it would take maybe roughly 100 million years for the Earth to cool down. You have liquid water on the surface, possibly volcanic island arcs that would give you surface land environments that could be relevant. Finding the right, after you get to that stage, maybe it takes millions of years to get just the right geochemical environment. There's very interesting, actually, new ideas that moderately large impacts could result in transient reducing environments that would allow for the synthesis of sort of a Miller-Urey environment, reducing, make a lot of cyanide, cyanoacetylene. Then you have to accumulate these molecules in stable reservoirs, like ferrocyanide salts, for example. And one of the open questions is how many times do you have to do that, right? Build up a reservoir of some starting material or intermediate. It has to survive for some point and then get processed or transmitted to a new environment for the next reaction or a couple of reactions. Then build up a new environment, say a crystalline intermediate like ribosaminoxazoline. If you have to do that several times in a row, right, it could take a long time and it could be extremely low probability. I think that's one of the reasons why the probability for the origin of life could, I'm not saying it is, but could in principle be extremely low. As you get closer and closer to the actual formation of life with reactive nucleotides, everything has to happen faster and faster, right? Because these molecules are more reactive, they're less stable, RNA degrades. I think once everything was just right, life just took off in a very short time. You had a question down here?
So many thanks for your interesting talk. I would love to see the experiments on the start of Darwinian evolution going on. But for that, I need you to answer positively to a different kind of question.
Okay.
Could aging be cured? Do you think that aging might be cured at some point in the near future, preferably?
I mean, if aging could be cured, I would be more optimistic about living long enough to see the beginnings of Darwinian evolution in these experiments. I do think that we will, aging is so complicated, right? There's so many things that go wrong. So it's not like there's one magic cure for everything, right? But I think, piece by piece, the different individual problems will be at least partly solved so that people can live healthy lives for longer. So obviously everybody wants that to happen, and that's why so many people are working on it, but I think that's also going to lead to the worst social problems you can imagine, so probably not enough people are thinking about the consequences of this happening.
I do have more questions. So when you mentioned that colder is better, I guess that there is a breakdown to that particular rule, right? So eventually, I guess that there is a limit of at which you lower temperature that everything goes maybe less errors, but more slowly. So there is actually a kind of a compromise.
What's the optimum there? Right. You could go a little lower, but having a salty solution, for example, not very much. Another really interesting open question is, could you actually do this in a kind of uniform, like isothermal environment, or do you need fluctuations in temperature? We don't really know. It seems simpler to imagine things working if you have fluctuating temperatures. So that's why I like these volcanic areas, right? You can have temperature fluctuations on very rapid time scales. But again, we don't know. We have to see by experiments what works and what doesn't.
And related, well, sort of related to that, do you know in your experiments what is the role of having or not having oxygen around?
None of the chemistry that I described here is sensitive to oxygen. It's the earlier steps that are very sensitive to redox state. A lot of the chemistry that's driven by iron and sulfur chemistry, the redox states are important, and that's why life like this could never start today, right? The earlier chemical steps absolutely could not happen. Once you get to this point, it's not really affected very much. Really.
Thank you very much, Jack, for your nice lecture. We have assumed that there are some clean or neat transitions in the early evolution of life, like RNA came first, then came proteins, afterwards came DNA. But it seems that systems chemistry is showing us something more, not so neat, not a little bit more dirty. For instance, the simultaneous appearance of RNA and DNA. What is your opinion about that? I think it's much more complicated than step by step, probably, but much more interesting.
Yeah.
What do you think?
Yeah, so there definitely are ideas about RNA and DNA maybe emerging together and sort of having some bifurcation. There are some chemical processes that can lead to deoxynucleotides being formed in the same reaction mixtures as ribonucleotides. So it's certainly possible that the first polymer was a heterogeneous mixture. And of course, once you have polymerases, they can be specific for one or the other. So that's, again, we've worked on that a little bit, not very much. That's an interesting question. Maybe even more interesting is the question of the primordial roles of amino acids and peptides. What did the first peptides do that were useful, that led to selection for coded translation? We've worked a little bit on roles of just simple amino acids, which can be spontaneously amino isolated, so attached to the ends of RNA molecules. They can facilitate loop closing reactions. So we think that amino isolated RNAs had to have been doing something useful before the ribosome, right? You couldn't evolve the ribosome, which uses amino isolated RNAs as a substrate, unless they were already there. Which raises the question, what were they doing before they were being used to make peptides? But there's also the question of what were the roles of the first peptides? And again, there are ideas, but not enough experiments yet. So yeah, I think all these things, ways of bringing in more complexity a little bit at a time so that we understand what we're doing, not just throwing everything together it's the way to go.
Do you have data on non-canonic genetic alphabet? As in, we had the arrows and such in A, G, C, and U, but we don't have on, well, I didn't see in I, N, or the bases with sulfur.
Yeah, no, we haven't really gotten to that yet. We're trying to do this. There's some technical problems because when you use reverse transcriptase to copy the modified nucleotides, you want to be sure that it's copying them accurately. And so we're still working out some of the technical details for that. But yes, a very important next step for us is to look at the accuracy with these modified nucleotides. So yeah, we're trying.
Okay, thank you.
And we have time for a last question.
So thank you very much. Considering that it turned to be so important environment where the reactions take place, do you know if the compartmentalization that the vesicles itself could activate the reactions somehow? I mean, what about to consider the primordial amphipathic molecules with the capacity to form vesicles? Yeah. And if that could have had, not only because of the compartmentalization, but also if chemically can help to some of the process.
Yeah, no, that's a really interesting question. We have tried a few things to localize the replication chemistry to the surface of the membrane, because you would think it might make it faster, right? Because it's a two-dimensional surface instead of three-dimensional search space. Everything we've tried there has basically not worked at all, but maybe we just didn't try it the right way. So it's still, I think it's an interesting idea. It is unfortunate that the chemistry that gave rise to the first amphiphilic molecules, like fatty acids, is the least understood. I mean, it's kind of crazy. We know how to make nucleotides, but we don't know how to make a fatty acid, right? And so, yeah, I'm hoping that some of our friends in chemistry will figure this out. I mean, there are some interesting ideas, but it's been very challenging. So it would be very nice to know more about how to make those first molecules and then how they would interact with the whole system. So far, we encapsulate RNAs. We can do copying chemistry inside, but it's more of a passive encapsulation, not being actively helped.
I think that we are, thank you, but before a big clap or round of applauses, I would like to rescue some of the words that you mentioned when finishing your talk about the need of environments in the labs that are diverse. With that, I would like to recommend to all the audience to a publication that Jack published in April this year, I think, and the title went like, I am an immigrant Nobel awardee and I don't recognize this country. It's a wonderful piece, and it's very important that we speak up, and we also would like to thank you for that. So thank you for your talk and your compromise.
Thank you.